So now we're going to have as a second presenter, Leon Mugenzi. Leon, could you start sharing your presentation? Meanwhile, Leon obtained his Bachelor of Science in Biochemistry and a Molecular Science degree in Molecular Diagnostics Science, and his PhD in Biochemistry and Molecular Biology in the University of Buea. His research interest centers around understanding mechanisms of insecticide resistance in malaria vectors. His current research is focusing on transcriptional regulation of genes conferring insecticide resistance in Anopheles funestus, a major malaria vector in Africa, and developing molecular tools to monitor insecticide resistance spread in field mosquitoes. I hand it to you, Leon.
Okay, thank you, Emma. Can everybody hear me?
Yes, yeah, we can.
Okay, so thank you, and thank you for giving me the honor to present my research. What I've been doing on is a continuation of my PhD work. So, and I'll be presenting on the impacts of a 4.3 structural variant on the gene regulation, on the gene expression and the insecticide resistance phenotype in the malaria vector Anopheles funestus. So as an introduction, we know that significant progress has been done in reducing the burden of malaria, but with a lot of cases that has been reduced. We had 1.5 billion malaria cases adverted and 7.6 million deaths adverted between 20 and 2015. And most of the progress has been thanks to long-lasting insecticide-treated nets, which have adverted 68% of those cases. And but from 2015 to now, we have seen a plateau in the progress, and most of it is attributed to the Plasmodium that is like escaping the detection from rapid tests. We have drug resistance. We also have recently the COVID-19 that has disrupted some of the interventions, and we also have the insecticide resistance. So from my work, I'm focusing more on insecticide resistance, and we know that previously we had bed nests that were impregnated with insecticides, and when the mosquito came in contact with that, they were sensitive and they were dying. But nowadays we are seeing reports of more mosquitoes that are becoming resistant to insecticide. And as you can see on your right, you have a picture showing the evolution of insecticide resistance in red dots to one of the insecticides used in bed nets impregnation, the pyrethroids. So this insecticide resistance involves the selection of an inheritable characteristic. So it is due to a DNA variation. And most of the research have focused on identifying and understanding how the genes and specific genetic variants can contribute to insecticide resistance. This knowledge is quite important and necessary to track and anticipate the cause of insecticide resistance for the national malaria control programs, and also for understanding the impact that this insecticide resistance can have on malaria transmission. And a previous work that we have done, we have shown how having some molecular markers could help track and also show how most of this resistance could impact bed net efficacy. And so, and for our work, previous study has been focusing on the coding region, so mostly on the genes, and also on SNPs, and also on small indels. But we have little information on the no coding regions, which is involved in the regulation, and also the role of structural variants such as large deletions, insertion, duplications, or chromosomal inversion. So we need more studies are needed to investigate their role in insecticide resistance. And for our group, we focus more on Anopheles funestus, even though we do some work on Anopheles gambiae too. And in the previous work published without 2020, we succeeded to identify some of these structural variants, which included copy number variation, gene inversions, and also transposable elements in the genome of funestus. And we also have succeeded to show one of the structural variants is 6.5 kb, which was inserted in a P450 cluster, and we showed that it was also driving insecticide resistance in the malaria vector. But this structural variant was limited mostly to Southern Africa. It was not present in the West and Central Africa. So still on our search of things that could explain insecticide resistance, the bioinformatics in our groups were focusing on PUSEC data that we had for the Anopheles spinaceus across Africa. And in screening the ones of Cameroon and Uganda, they found a structural variant that was found at high frequency in Uganda, almost close to fixation, while in Cameroon, using the data from 2014, it was present, but at a low frequency. So knowing the position of this structural variant, we succeeded to amplify this. And usually you expected in the reference genome to have 1 kb size, but when with the structural variant, it was shown to be 5.5 kb. So when we succeeded to amplify this region and we sequenced and we found out that this region contained open reading frame of 2 retrotransposable elements. And this region is good to know that it was also present in another location of the Anopheles spinaceus genome. So we proceeded to try and design a simple DNA assay that could permit us to distinguish between mosquitoes that have the structural variant and the ones that don't have. So it's just based on 3 primers. So 2 that are flanking the insert, such that if a mosquito doesn't have, you have a 600 base pair, while with the insert, you have a 900 base pair. So we validated this, and then we tried to look at, survey its distribution across different locations in Africa. And what we noticed is that it was present, it was absent in West Africa, present in Central Cameroon, and East Africa, Kenya, and Uganda, while it was, and Tanzania at a low frequency, while it was absent, complete absent in Mozambique, Southern Africa. So here we have the allelic frequency and the genotypic frequency, and you can see that the double homozygote for the structural variant at a high frequency. So then we focused on Cameroon, since we are located in Cameroon and where the structural variant was present. We tried to check for locations across the country where Anopheles spinaceus population have been collected, and we found out that it was present at a very high frequency, approaching 95, almost close to fixation. But there was one location, Gunugu in the north, where it was not almost fixed. We had at 60%. And the next question was to try and see if it has always been like that, or we have had a selection over time. And we are lucky to have samples dating from 2016 from previous years for the different locations. So we used Mibelon, Tibati, and Gunugu. And what we found that using Mibelon in 2016, the structural variant was present at a quite low frequency. But from 2018 going to 2020, we have a very high selection, going almost to fixation. And this was also a similar pattern that was observed in another location, Tibati, and also in Gunugu. There was a selection, but it was less than in the other, what was observed in the other location. So using the location where this structural variant was not fixed, we tried to identify what could be driving its selection. And one of the main thing was insecticides. So we tried to see if it was not selected by the use of insecticides. And for that, we focused on samples from the Gunugu location. And we had samples that had been exposed to pyrethroid delta-metrine. And we had 60 minutes following the WHO protocol. And we had mosquitoes that were alive and dead. So when we genotyped the structural variant among the alive and dead, we found that it was strongly associated with the ability of mosquitoes that were alive, while the ones that were dead were mostly lacking the structural variants. Both at the allelic frequency and also at the genotypic frequency, we noticed that being a homozygote for the structural variant, you had a higher ability to survive. So, and another way of answering this, since most of the location had a fixation of this structural variant, we did a genetic cross using field mosquitoes that are homozygote for this, and the susceptible lab strain fang, which is quite susceptible and doesn't have any of this structural variant. So we did a crossing and we allow it to intercross to the F3 such that we could have all the 3 genotypes present. Then we did bioassay using delta-metrine at F3, and we also noticed the same pattern as what we had observed in the field. So we noticed that most of the alive had the the 4.3 kb structural variant, while most of the death lacked it. And it was the homozygote, and what we also noticed that the homozygote for the structural variant were all alive. Heterozygotes, they were among the alive and the dead. So we also checked if this could be, the similar pattern could be observed using PermaNet. So we used the cone assay, where mosquitoes are exposed to a piece of the PermaNet instead of using paper impregnated with insecticide, like for the WHO test. So, and for that, we used also a PermaNet that is impregnated with delta metrin, so PermaNet 2.0. And for that, we exposed it 3 minutes, and then we had the alive and the dead mosquito, and we tried to genotype. So upon genotyping, we also noticed that mosquitoes that were homozygote for the structural variant were among the alive, while the ones that were without the structural variant were among the dead. And this was observed at the genotypic and the allelic genotype. So still trying to understand what could, how this structural variant could contribute to insecticide resistance, we still used our genetic cross, and we try to see how this structural variant would impact the expression of genes that are neighboring this structural variant. And for that, we choose P450, Sub6P5, Sub6P9B, and Sub6P9A. And then we did a qPCR using homozygote for the structural variant, which are in yellow, heterozygote, and the ones with homozygote without the structural variant. And what we noticed is that the two P450 Sub6P9A and Sub6P9B were overexpressed in the homozygote genotype, but not in the heterozygote, showing that you need 2 copies to overexpress these genes. And these genes have been previously implicated in insecticide resistance in Southern Africa. So maybe one way how this structural variant is driving resistance might be through the expression of these neighboring genes, but we still need to carry out some further functional validation to show its impact on gene regulation. So having this marker, we also try to see if it could be associated with Plasmodium susceptibility. And what we did for that, we had samples that were collected in Obut 2016. And this location, we have high Plasmodium infection, and also the in mosquitoes, and also this structural variant was not yet fixed in 2016. And so we genotyped the uninfected and the infected mosquitoes, and we noticed that most of the homozygote for the structural variant were among the uninfected, while the ones that didn't have were among the infected. So showing that maybe there's a possibility that the upregulation of P450s, which is an adaptation to insecticide resistance, can also make resistance more resistant to pathogen. But further studies are needed to really understand to understand the mechanism behind this. So as a conclusion, we can see that this structural variant is present in Central and Eastern Africa and is under strong selection. We also found an association with the resistance to type 2 pyrethroids, detametrin. And also we found an association with the increased expression of 2 resistant genes, type 6P9A and type 6P9B. And we also find a negative association with the Plasmodium infection in the Anopheles phoenicus mosquito. So I would like to acknowledge the PI of the project, Professor Charles Wanji, Dr. Jacob Riveron, and the bioinformatician Garrett Widal and Jack Earn, and also some of the PhD and master students that worked on this. I'd like also to thank our founder, the Wellcome Trust. Thanks for your attention.
Great. Thank you very much, Leon, for such an exciting presentation. So we have some questions in the Q&A. So let me start reading. So if you remove the drug pressure, are these structural variants revert back to the normal?
Thanks for that question. Usually this will be the case in case it was not yet close to fixation, but in the location where we have fixation of this structural variant, it might be difficult to revert. But in localities where it is still at 60 or 50%, maybe removing the insecticide pressure might reduce the frequency. But we will try that in the lab.
Great. Thanks.
So this is a purely speculative question, Leon. In areas where this is fixed, would you recommend that they change the insecticides?
Yes, definitely. We need to change the insecticide. Yes.
Also, I'm wondering if there are any studies about like the use of herbicides, like the glyphosate tolerant insecticides or the glyphosate tolerant crops in the area, and have you seen any? Is there any study about these any correlation of these mutations happening where these fields are being spread with this herbicide?
Good point. So far, we have not looked at herbicide. It's true that most people have been focusing on pesticides, but I think we could explore also the contribution of herbicides to the development of resistance, but it has not yet been done.
Yes. Thanks. There is another question. Do you know if this resistance occurred de novo or are transmitted from one location to another, especially between countries?
Okay, thanks. That's a very important question, and we are still trying to answer that by sequencing the region and seeing if we have similarity. Because at the first point, we hypothesized that it might have migrated from Uganda, since it was almost very high in Uganda and Kenya and absent in Cameroon. But we need to sequence and try to see if it's a de novo or a migration issue.